hello everyone, and welcome to this audio podcast. This audio podcast as part of the probe series. This audio podcast is part of the Storm and Smash Up. This audio podcast is part of the Consular and Shout Out, and this audio podcast is part of the Specials and a Storm Robotics production. Through this audio podcast and in as part of works by a science student and Bohtha that developed some fantastic works looking at looking in on at work in on cellular localization of breast tumor kinase or BRK and protein tyrosine kinase 6 in tumor development and in on across different forms of cancer. This audio podcast will be in on BRK activities and locations. So initially, BRK was cloned from melanoma cancer cells. BRK phosphorylates many nuclear and cytoplasmic substrates such as ribonucleic acid binding proteins or RNA binding proteins, and signal transducers and activators of transcription with molecules which activate growing factor receptors and gene expression regulators are form sort of part of the form part of the sort of some of the main components of BRK, and yeah, all form sort of the main components of BRK or around the tumor, this particular tumor kinase, and the location of BRK is important because the cellular environment can limit its activity when the substrates are limited in cytoplasm or nucleus. BRK is also different in tissue type shows in BRK is also different and different tissue types also show different roles and react with different substrates. BRK function depends on its cellular location, tissue, and on the substrates it interacts with. The RNA binding protein, which the RNA binding protein, or where there is RNA binding protein, there is a localization in amongst nuclear bodies of cancer cell lines and is seen in nucleus during mitosis. The particular RNA binding protein is also involved in mRNA messenger RNA metabolism and is present throughout transcription until exportation from the nucleus. BRK negatively regulates the function of and in around the RNA of and in around a particular binding RNA binding protein and mammalian protein in nuclei or nucleus. Furthermore, BRK phosphorylates in around two other additional sort of binding sites, Y31 and Y118, creating sort of binding sites for additional sort of linkages for example some studies or some investigations have shown that phosphorylation of particular particular components such as TRYR1105 increases increases P190, which is linked with additional components, elements and components, leading to inhibition, but also activation of additional cells or cell elements, and activation of particular protein switches on some elements of and around particular targets, where particular cells are targeted, which then becomes more important for cell division and proliferation in amongst cancer. And then there is sort of particular sort of transcription factors, such as cytokines cytokine signaling and growth factors, and where cytokine and growth factors then prompt particular transcription factor, being STAT3, for example, which is a transcription factor which regulates biological processes like mitochondrial metabolism and is located in the endoplasmic reticulum and in cytoplasm. STAT3 is also shown in amongst nuclei and is a major factor for survival and cell growth, tumor cells, reducing signaling pathways to slow down cancer development. STAT3 is also used or has been used as a form of pharmacotherapeutic target for cancer, in particular forms of cancer. STAT3 inhibitor in amongst literature has been evidenced where the inhibitor tuzanidine is utilized or has been utilized as a treatment for osteosarcoma. In addition to signaling for growth factors, calcium also activates BRK in cell membrane during keratinocyte differentiation in the epidermis. Keratinocyte differentiation then controls the action of tyrosine kinase and control proliferation of and in around breast cancer cells. So that draws this audio podcast to a close. This audio podcast is part of the Probe. This audio podcast is part of the Ztorm and Smash Up. This audio podcast is part of Consular and Shout Out. This audio podcast as part of the Specials and a Ztorm Robotics production.